Greetings to everyone. Welcome back once again to this educational channel on biology. In this short video, we'll be discussing activity 9.1, 9.2 and 9.3 in the Form 4 Biology KSSM syllabus. And these activities are to investigate the digestion of starch, proteins and lipids in food samples by enzymes. So let's get started. Right, activity 9.1. The aim is studying the digestion of starch in a food sample. Page 147 of your Form 4 textbook. Now, problem statement. What is the effect of amylase on starch? So amylase is the enzyme and starch is the substrate, okay, which amylase acts on. Our hypothesis. The enzyme amylase hydrolyzes starch to form a reducing sugar, which is maltose actually, okay. Now, variables. Your MV, manipulated variable, is the presence of amylase. So one test tube will have amylase and the other one will not have any amylase in it. Now, responding variable is the presence of the reducing sugar. So we're going to test the presence of the reducing sugar after some minutes, right? So we're going to test for the presence of reducing sugar such as maltose. Then the fixed variable, temperature of water bath, is at 37 degrees Celsius, which is the optimum temperature for enzyme reaction to occur more quickly. Concentration of starch suspension is also a fixed variable. So we use 1% starch suspension. Okay. So materials, 0.5% amylase solution, 1% starch suspension. Iodine solution is to test for the presence of starch. Okay. After we have left the apparatus and materials for a while, after we leave the apparatus set up for a while, and then Benedict solution, and then distilled water. So Benedict solution is to test for the presence of the reducing sugar, such as maltose. Now, apparatus that we need: tripod stand, Bunsen burner, wire gauze, 500 ml beaker, test tube holder, thermometer, stopwatch, test tube dropper, glass rod, and measuring cylinder. So let's go on to the procedure. Two test tubes are labeled A and B. 3 ml of 1% starch suspension is added to each test tube. But the difference here is test tube A is filled with 3 ml of 0.5% amylase solution, whereas test tube B is filled with 3 ml of distilled water. So in test tube B, there's no amylase solution. Instead, the amylase solution is replaced with distilled water. Both test tubes are placed in a water bath with a temperature of 37 degrees Celsius for 10 minutes. So this is the optimum temperature. 37 degrees Celsius is the optimum temperature for the highest rate of enzyme reaction. Now, after 10 minutes, okay, we only put the test tube in the water bath for 10 minutes. So after 10 minutes, 2 ml of the solution is removed from test tube A and put into a different test tube. So we're going to test for reducing sugars using the Benedict solution. Okay, so this is called the Benedict's test. 3 drops of Benedict solution are added to the test tube containing the sample of solution from A, which is then heated in a boiling water bath for 1 minute, and the color of the content is recorded. Okay, so we must heat the mixture of Benedict solution and the sample solution in a boiling water bath for 1 minute in order to see, and then after that only we note the color of the content. Okay, whether it's brick red, whether there's a brick red precipitate, or whether it remains blue. Okay, now 2 drops of iodine solution is added to the remainder of test tube A. The color of the contents is observed and recorded. This is the test for starch, okay, using iodine solution. Then repeat steps 5 and 6 for test tube B, okay, because we want to test for the presence of 2 substances, starch and also reducing sugars, in both test tubes. Now the Benedict's test for reducing sugar. So let's revise this. Add 2 ml Benedict's solution to 2 ml of sample solution in a test tube. Note that we add equal volumes of Benedict solution to the sample solution. If you add too little Benedict solution, sometimes the results are not clear. Now, number 1, heat the test tube in a boiling water bath for a few minutes, right, after you have mixed the 2 solutions together, right? So the color is blue in the beginning due to the Benedict's solution, right? It may be blue. So if the reducing sugar is present, the color will change. Okay, the solution changes color from blue to green to yellow and orange, and then finally a brick red precipitate is formed. Okay, so if it changes to green, yellow, or orange, it still means that there is reducing sugar present, just that it is in smaller amounts. If it's a brick red precipitate, it means that there's a lot of reducing sugar present. Okay, so if reducing sugar is absent, then the solution does not change color, remains light blue, right? Now, these are some of the color changes that you may see or observe when you carry out the Benedict's test for reducing sugars. As you're heating the mixture containing the Benedict's solution and the sample, the food sample, for example, it will change color from blue to green to yellow, then to orange, and finally to brick red, to form a brick red precipitate. Okay, so even if it is a yellow or orange color, it means that there's some reducing sugar present. Maybe not a lot, not so much. Now, the use of Benedict's test is to test for the presence of reducing sugars such as fructose, glucose, galactose, maltose, and lactose. This is the apparatus set up for our experiment to study the digestion of starch in a food sample. Okay, so the temperature is fixed at 37 degrees Celsius by using a water bath and a thermometer to ensure that the temperature remains constant. Okay, so this is our constant variable or fixed variable. So in test tube A, we have starch suspension and amylase solution. In test tube B, we have starch suspension and distilled water. Okay, then we leave this apparatus set up for 10 minutes. Finally, after that, you want to know whether digestion has occurred in test tube A or B and B, right? Whether digestion of starch has occurred. So we are going to test for the presence of starch and reducing sugars, which is maltose in this case, right? In both test tubes. That means for the mixture in both test tubes. Right, let's look at the results of our experiment. Test tube A contains starch, suspension, and amylase solution. So after 10 minutes, when we test it with the iodine test, we found that the mixture was brownish yellow. So this brownish yellow color is actually the color of the iodine. That means there is no reaction to the iodine test, meaning that starch is absent. Okay, there's no blue-black color like this below here. So it shows, if it's brownish yellow, it shows that starch is absent. That means if starch was here, was there at the beginning, was present at the beginning of the experiment in the test tube, now the starch is absent. So it shows that the starch has been digested. Okay, so it should be digested to form maltose, right? So we are going to do the Benedict's test to test whether reducing sugars such as maltose is present. Okay, so take a bit of the mixture from A and then add the Benedict's solution to it, and then after that heat it up. Okay, then we get a brick red precipitate. This shows that reducing sugar, maltose, is present. Okay, so starch is absent, maltose is present. That means digestion has occurred. According to this equation, starch plus water, okay, in the presence of amylase here, which catalyzes the reaction, we get maltose. Okay, so this reaction occurs in test tube A, and all the starch has been digested and maltose has been formed. That's why we get the brick red precipitate. Okay, and there's no longer starch in the test tube anymore. So the iodine test shows a brownish yellow color. Now, for test tube B, right, starch suspension and distilled water, after 10 minutes, the starch should still be there because it's not digested. There's no enzyme in test tube B. So we get a blue-black color, okay, like this, showing that starch is present and no digestion has occurred. Okay, then when we do the Benedict's test on this mixture, okay, and on the second portion of this mixture from test tube B, we get a final result of a blue solution. That means this blue solution is the color of the Benedict's solution, right? So there was no change at all. There was no brick red precipitate form. So this shows that no reducing sugar such as maltose was formed. Alright, so take note of these colors and also take note how to write the results. If the result is negative for starch, we write that the iodine remains brownish yellow in color, or the mixture remains brownish yellow. But if there is a positive result to the iodine test, we can say that the mixture or the substance turns blue-black, okay, to show the presence of starch. Okay, and for Benedict's test, the positive result is brick red precipitate, showing the presence of reducing sugars. And if there's no reducing sugar, then the solution remains blue, okay, like in the Benedict solution. Next, studying the digestion of protein in a food sample. Right now we're talking about protein. So what is the effect of pepsin, the enzyme pepsin, on protein? Our hypothesis: the enzyme pepsin hydrolyzes proteins in albumin suspension, okay, into polypeptides. So albumin is something like the white of the egg, right, which contains proteins in it, okay, and our hypothesis is that the pepsin will break down the protein to form simpler molecules called polypeptides. All right, so our manipulated variable is presence of pepsin, and responding variable is clarity or turbidity of mixture after 20 minutes. Now, turbidity means the cloudiness of the mixture. Whether it's still cloudy, like when you put some milk, drop a few drops of milk into water, it turns cloudy, right? Whitish and cloudy. Okay, so we are going to see whether the mixture becomes clear or is still cloudy after 20 minutes. Okay, so actually the cloudiness or turbidity of the mixture is due to the albumin suspension at the beginning of the experiment. But will it still remain after 20 minutes? That's the question. Now, will the solution still be turbid? Fixed variable, temperature at 37 degrees Celsius, concentration of pepsin solution, and the presence of the dilute hydrochloric acid to create acidic conditions, because pepsin works best in acidic conditions with a pH of 2 to 3. Right, materials: albumin suspension or egg white, 1% pepsin solution, distilled water, 0.1 m dilute hydrochloric acid, apparatus, test tube, measuring cylinder, 500 ml beaker, water bath, and a dropper, and thermometer and watch. Right, let's look at the procedure to study the digestion of protein. An apparatus setup is prepared as follows. Test tube A contains 5 ml albumin suspension, 1 ml of 0.1 m hydrochloric acid, and 1 ml 1% pepsin solution. Test tube B contains the same contents except that the pepsin solution is replaced with distilled water. Okay, then both test tubes are placed in a water bath at the optimum temperature of 37 degrees Celsius. The condition of the mixtures in both test tubes are observed at the beginning of the experiment and after 20 minutes. So this is the apparatus setup, right? We have a water bath, and it's at the temperature of 37 degrees Celsius, which is the optimum temperature for the fastest enzymatic reaction. So in test tube A, we have albumin suspension, hydrochloric acid, and pepsin solution. So pepsin is the enzyme here. And then in test tube B, there is no pepsin solution. The rest of the contents are the same, but there's no pepsin solution. Instead, the pepsin is replaced with distilled water. Okay, so there's no enzyme in test tube B. All right. Right, so at the beginning, both these test tubes contain the albumin suspension, which is turbid or cloudy. Okay, so it looks a bit opaque. It's something like when we add some milk, liquid milk into a cup of water, it'll turn cloudy, right or not? So it's like whitish and cloudy, a bit cloudy, because due to the albumin suspension, right? So let's see what happens after 20 minutes of leaving these two test tubes in the water bath. Right, so after 20 minutes, we can see here the solution has become clear. So the turbid albumin has been digested by the pepsin solution to form a clear solution, which is a solution of polypeptides that is soluble in water. So it's a clear solution. So the albumin has been digested by the pepsin to form polypeptides. Whereas in test tube B, there's no enzyme here, so the albumin is still present. So the mixture here is still turbid or cloudy because the albumin is still there and it's not digested. It forms the turbid or cloudy suspension here, right? So that means for this experiment, to detect the presence of protein, we look at the turbidity or clarity of the mixtures or the solutions in A and B to detect whether the albumin is present. So, okay, so if albumin is not present, then it forms a clear solution, right? Especially when it's been digested to form polypeptides, right? And here, because albumin is present, there is still a turbid or cloudy suspension caused by the albumin. Let us look at the results of our experiment here. So in test tube A and B, at the beginning of the experiment, both were turbid or cloudy due to the presence of albumin. After 20 minutes, after leaving the apparatus aside for 20 minutes, when we look at test tube A again, it has turned clear, no longer cloudy or turbid. So this shows that this reaction has occurred, which is protein plus water, okay, and this reaction is catalyzed by enzyme pepsin, has become polypeptides. Polypeptides are soluble in water, and then it forms a clear solution, okay, whereas albumin is turbid and cloudy, forms a suspension in the solution. All right, so when the turbid solution becomes clear, it means that polypeptides are formed and digestion of protein has occurred. That means pepsin has digested or hydrolyzed the protein to form polypeptides. Lastly, studying the digestion of lipids in a food sample. Okay, activity 9.3, page 149 of the Form 4 textbook. Problem statement: what is the effect of lipase, the enzyme lipase, on the substrate lipid? So lipase hydrolyzes lipids into fatty acids and glycerols, right? So our variables are manipulated variable, presence of lipase, responding variable, time taken for the phenolphthalein indicator to turn from pink to colorless, and fixed variable is the temperature of the water bath again, it's at 37 degrees Celsius, the volume of cooking oil used. Okay, so cooking oil is the source of our lipids. Now, cooking oil, materials are cooking oil, 0.2 m sodium carbonate solution, dishwashing liquid, distilled water, the enzyme lipase, phenolphthalein indicator. Apparatus, we need 2 test tubes, test tube rack, water bath at 37 degrees Celsius, 5 ml and 1 ml syringes, dropper, stopper, and stopwatch. Right, let's look at the procedure. 2 test tubes are prepared and labeled A and B, just like for the other 2 experiments. In each test tube, okay, each test tube A and B is filled with the following. Okay, these are the common items in each test tube. Okay, 2 ml of cooking oil. Now this is our lipid. All right. Now we can also use full cream milk, which contains lipids, in place of cooking oil. Then 1 ml of 0.2 m sodium carbonate solution. Sodium carbonate solution is to produce an alkaline condition of about pH 10 to 11. All right, it is an alkaline. And 1 ml of this dishwashing liquid, it acts like bile salts. Okay, it emulsifies lipids by increasing the surface area of lipids for faster hydrolysis by enzyme lipase. Okay, so it's not the enzyme, but it acts as a emulsifier. So both test tubes are closed with stoppers, and the 2 test tubes are shaken vigorously after adding the dishwashing liquid to emulsify the lipids. Then we add 3 drops of phenolphthalein indicator to each test tube, and then the test tubes are shaken again. Now, so the color of the mixtures in both test tubes should turn pink due to alkaline conditions. So this phenolphthalein indicator is pink in alkaline conditions, right? And colorless in acidic conditions. Okay, but because of the sodium carbonate solution, it will turn pink, because sodium carbonate is an alkali. Now, then in test tube A, we add 1 ml of lipase enzyme, the enzyme lipase. In test tube B, we add 1 ml of distilled water to replace the lipase. Okay, so test tube B is our control. Both test tubes are placed in a water bath at 37 degrees Celsius, which is the optimum temperature for faster enzymatic reaction. Then time taken for the phenolphthalein indicator to turn from pink to colorless is recorded. Will the phenolphthalein indicator turn from pink to colorless? Now, when it turns colorless, it means that the condition is below pH 8.3. It could be below, it could be still slightly alkaline, and then it could, or neutral or acidic. Then it will turn to, when the conditions are like this, it will be, the indicator will turn to colorless, from pink to colorless. Here's the apparatus set up to study the digestion of lipid in a food sample. Now, at the start of the experiment, right, we have test tube A and B. Test tube A contains cooking oil, sodium carbonate solution, which is alkaline and is pH is around pH 10 to 11. Then we have 1 ml dishwashing liquid to act as the emulsifying liquid to break up the fat droplets, right, in the cooking oil, or the lipid droplets in the cooking oil. Then we have phenolphthalein indicator, which turns pink in alkaline conditions. Okay. Now think of the word pink, P-I-N-K, alkaline. Okay, there's a K here also. So phenolphthalein is pink in alkaline conditions, right? And then there's also the enzyme lipase added to digest or hydrolyze the lipids in the cooking oil. Okay. Now for test tube B, which is a control, all the contents are the same except for the lipase. There's no lipase in B, but instead there is distilled water. Okay. Right. Let's look at the next. What happens after 10 to 20 minutes? All right. Right. After 10 to 20 minutes, we get this result. We see that in test tube A now, the phenolphthalein has turned from pink color to colorless, whereas in test tube B, the phenolphthalein remains pink. Okay. So what happened in test tube A? Now the enzyme lipase in test tube A has digested the lipids to form fatty acids, and these fatty acids neutralize the sodium carbonate in the test tube A, which is an alkaline. So it causes the condition of the mixture to turn from to be reduced in pH from pH 10, the pH lowers to pH 8.4, 8.3. Okay, and then below 8.4, 8.3, the phenolphthalein will turn color already, right? And the pH may even go down to neutral condition and then acidic condition as more and more of these fatty acids are formed, okay, from hydrolysis of lipids by lipase. So below pH 8.3 or 8.4, the phenolphthalein already turns colorless and remains colorless in acidic condition, right? So think of this word, acidic, there's a C there, right? So colorless. So phenolphthalein is colorless in acidic condition, neutral condition, and below the pH of pH 8.3 or 8.4, right? Whereas for test tube B, there is no color change. The phenolphthalein remains pink as at the start of the experiment. Okay, so can you explain this result? Now, when distilled water is present here in place of lipase, there's no enzyme to digest the lipids, right? So there's no digestion of lipids and no fatty acids are formed. So the sodium carbonate that was present is still there, and it will create an alkaline condition because it's not neutralized at all by any fatty acids, right? So the phenolphthalein is in an alkaline condition, and it will remain pink. Okay, so there's no reaction here in test tube B. Okay, only in test tube A, where hydrolysis of lipids have occurred. Okay, hydrolysis of lipids by lipase has occurred. So the presence of the fatty acids is detected by the phenolphthalein, which actually detects the change in pH, okay, caused by the fatty acids. Okay, the change in pH from a very alkaline condition to less alkaline and neutral and more acidic condition, right? Then the phenolphthalein turns colorless. So when we record our results, it will be like this. Test tube A, the contents. We have all the other substances and then including 1 ml lipase, which is an enzyme. So time taken for phenolphthalein indicator to turn from pink to colorless in minutes. Now, this can depend on the type of cooking oil used and the freshness of this lipase, and also other factors. Okay, but anyway, here the time recorded is 8 minutes. It could be 5 minutes, it could be 8 minutes, it could be 10 minutes or 15 minutes. It depends, right? So for test tube B, which does not have lipase, but it has 1 ml distilled water in it, there is no enzymatic activity, so the phenolphthalein indicator remains pink even after 10 or 20 minutes or 30 minutes. It doesn't change color. So you must write down that it remains pink. So the reaction in test tube A is lipid plus water, then hydrolysis occurs using this enzyme lipase, and the products are fatty acids and glycerol. Okay, so these are formed. So lipase has hydrolyzed the lipids into fatty acids and glycerols in test tube A, and the fatty acids then neutralize the alkali sodium carbonate. It reduces the pH of the mixture and may even cause it to turn acidic. Anyway, the pH will be reduced below pH 8.3 or 4, and this causes the phenolphthalein to turn colorless in this condition, okay, of less than pH 8.4, or in neutral and acidic conditions, it will turn colorless. So let's look at this flow chart to understand the reaction that occurs. So lipase hydrolyzes lipids into fatty acids and glycerols in test tube A, and the equation is lipid plus water, all right, and with the help of this enzyme lipase, right, to catalyze the reaction, we get fatty acids and glycerol. Then these fatty acids neutralize the alkali sodium carbonate in the mixture. So by doing that, it lowers the pH from pH 10 to below pH 8.4, and it can even reduce the pH to neutral conditions and then to acidic conditions. So anyway, below pH 8.4 onwards, the phenolphthalein, which is pink at pH 10, will then turn colorless, okay, because the pH is below pH 8.4. Now, to recap the observation and results. Observation is that phenolphthalein indicator changes color from pink to colorless in test tube A, which contains lipase, but it does not turn colorless in B, okay, which has no lipase. Reason: in test tube A, lipase hydrolyzes the lipids in cooking oil into fatty acids and glycerol, and these fatty acids then neutralize the alkali sodium carbonate, and this changes the pH of the mixture from pH 10 to below pH 8.4, okay, and even to neutral and acidic conditions, okay, due to the fatty acids. So this causes phenolphthalein to change color from pink to colorless, right? So remember, pink in alkaline conditions, alkaline got a K there, so it's pink, okay, there's a K here also. Colorless condition occurs in acidic conditions, for example, so acid AC, and you can see there's also a C in the word colorless. So in acidic conditions, the phenolphthalein is colorless in neutral and acidic conditions. So test tube B is a control experiment. It does not contain any enzyme lipase. There's no digestion of lipids, no production of fatty acids. Thus, the alkaline sodium carbonate is still present, not neutralized, and phenolphthalein remains pink in the alkaline solution. Right, that's all for this lesson. Thanks for viewing, and all the best for your test, all right, practical test.